Okay, so we're a little bit ahead of time, and so I can talk a little more slowly than I usually do, I guess. Thank you, Zach. And let me see. I want to talk about how this all started. I had one weird thing leads to another. I was supposed to go to med school right after college, and then something happened, and I had to take a year off, which turned out to be a great experience because I ended up getting a job in a research lab doing virology research, which got me very interested in research, which I didn't know I was going to be interested in. So I went to medical school and then decided to go to neurosurgery. And I was like, how do I combine research and neurosurgery? Fortunately, UCSF had a program that was really unique in that they really wanted people to get interested in research. We have another UCSF graduate here. And so my training at UCSF was theoretically in one of the world's top places, and I remember going to my first job at UAB and having a glioblastoma patient, and the family came up to me and said, "What causes this?" And I was thinking, as I was saying, we don't know. It's like, well, I guess I'm we now. I'm one of we, because I was at the best trained institution, and we really don't know. And then I started thinking, how many of us actually think about that more than just what causes it? We don't know. And I sort of happened to start thinking about unconventional things. And then I went to a lecture by a guy who's actually from UCSF, lived down the street from me in San Francisco, Jay Levy, who was one of the co-discoverers of HIV. And he gave a talk at the AACR meeting that I went to in one of those 7:30 a.m. breakfast symposia that there were about 3 people at, sunrise session. And he said something that sort of really got me thinking. He said, I'm going to talk about cancer, and everyone, as you know, will say cancer is a genetic disease, but I think that viruses could be involved. And he said there was an old colleague of mine who used to say, viruses don't cause cancer, cancer causes viruses. So he went on to explain what he meant by that, which is when a tumor cell turns into a tumor cell, it loses its endogenous, intrinsic antiviral response. And Dr. Jim Markert's going to give our keynote lecture later, and the whole foundation of cytolytic virotherapy hinges on the fact that a tumor cell, for some reason, permits viruses to infect it and replicate. And why is that? Well, we don't know every reason, but it has something to do with the intrinsic antiviral response that most cells have with interferon, but tumor cells somehow have an inability to deal with that. So that got me thinking, what kind of virus could potentially be involved in glioblastoma if one were possible? And this was with the background that, in developed countries, we know that about, 18% of cancers are associated with infectious diseases. And third world countries, it's almost 1/4 of cancers because there's such a greater prevalence of hepatitis-related hepatoma and stomach cancer. All of my kids have now been vaccinated for HPV, which 10 years ago was still kind of a radical concept, and that might eliminate cervical cancer from the United States. So, why couldn't other tumors be associated with malignancy? Other viruses. So in thinking this through, kind of came up with a short list of viruses that could be potentially associated with glioblastoma. And so I thought, okay, if I wanted to cause glioblastoma, what would I be? Well, I would be something that loves proliferating glial cells in the brain. I would be something that can mutate cells. I'd be something that causes a persistent infection, preferentially in stem cells, because we think stem cells are the cell of origin of cancer. I would be something that causes inflammation. I would be something that can dysregulate the immune system. And so I kind of from my med school memory, I think CMV does all that. So I asked this person in my lab, hey, can you do some immunostains for CMV and glioblastomas? And she's like, sure. So, before I get to that, how do I go back? Okay. So, before I get to that, there were a couple of other things that, after I started digging through the surface, that kind of seemed interesting. A picture this picture here shows that if you get CMV infection as a fetus in the second or third trimester, your brain essentially gets eaten alive, or can be, and you get calcifications and multinucleated giant cells. It's the #1 cause of mental disability in the United States. That's not often understood by people. It's the #1 cause of deafness in the United States. It's a huge public health risk. And also, when I was reading about, where does CMV live in the brain, if in the latent situation, there were studies in mice where they would infect mice with CMV with a beta-galactosidase gene in it, so it turned blue if the virus got reactivated. And if they take those mice brains out when the mice are apparently healthy and throw their sliced brain tissue into culture, the virus reactivates because there's no immune system. And where does it reactivate? It reactivates in cells that are the stem cells that live in the brain, specifically glial precursor stem cells. And those are the cells that we think glioblastomas derive from. And actually, there was a paper in Nature this year by a Korean group that almost definitively proves that. So, here's a virus that persistently infects the brain, can last for decades, and stem cells, and those are the cells we think cause glioblastomas. And so, when I first started talking to people, they're like, well, people should have tried these experiments where they put the cell on and turns into cancer, but that hasn't been done, so you're crazy, this doesn't happen. But the paradigm is really not that you add the virus and you get cancer. The paradigm for herpes viruses, which CMV is a member of, is more multifactorial. And sort of the prototypical cancer-causing herpes virus is Epstein-Barr virus. And this is a really wonderful review article that shows that EBV essentially works on the hallmarks of cancer, which were Hanahan and Weinberg's, I guess there are 10 of them now, hallmarks. If you get an EB infection, which 100% of us have, when you're a little kid, it gets into your white blood cells and all the viral genes get turned on. You may or may not have any kind of infectious, sequelae like fever. And then these CTLs, cytotoxic T cells, just shut this thing down. And for the rest of your life, you have EBV circulating in your white blood cells, your B cells, but it doesn't do anything because it's shut down. And those genes cannot be expressed in that setting. However, if you get AIDS or if you have immunosuppression, then those T cells don't function and these viral genes get turned back on. And guess what? We see B-cell lymphomas all the time in people that are EBV positive due to that fact. Or if you're in Africa and you have malaria and your blood cells are turning over at a rapid rate and a few of them happen to get MYC or P53 mutations, those mutations will sort of collaborate with and fill in the gaps plus EBV to cause Burkitt lymphoma, which is a EBV-related malignancy. So, these things don't happen in isolation, and if you put multiple hits together, theoretically you could get a cancer from a virus that persists. So, and then you throw in the fact that even back when I started, which is a long time ago, we knew that people with glioblastoma have defective immune systems. And the T cells in people with glioblastoma before any treatment are often completely anergic, which means if you give them the skin test with Candida, most people, you inject a little of this mold in your skin, and you get a big red welt on your arm. Glioblastoma patients don't get that. Why? Well, first of all, the tumor cells themselves are secreting multiple factors that are totally immunosuppressive, and I won't go through all of them, but we know the tumors secrete factors that can shut down T cells, like and induce PD-1 and CTLA-4, which Harvey spoke about. And then this is just a striking figure from one of the Duke guys' papers. When you look at the CD4 counts, which are the T cells involved in helper T cell mediated immunity, you see that patients with glioblastoma, the minute they walk in off the street, at least in this paper, and I think another paper shows a similar finding, their CD4 counts are around 200 or so, which is by definition the AIDS if you have HIV. So we know these patients have an immunological defect. So taking that all as a background in consideration, I started investigating this virus, and Over the last 15 years, this diagram, which is pretty busy, shows you that this virus encodes for over 150 different proteins, and all these proteins that are shown in green are proteins that can promote the hallmarks of cancer. So this virus is perfectly equipped to cause cancer by knocking out immune function, by driving tumor inflammation, by activation of metastasis pathways, by altering the cellular energetics, by genomic chromosomal instability, by cell proliferation pathways, and by evasion of tumor suppressor function. So if it has a chance, just like EBV, if you were to take away the T cells that suppress this thing, theoretically it could promote cancer. So what's the evidence? I mentioned that I had a person look at glioblastomas, and when she first did the studies, I thought that they were all negative, so I said, well, that was a stupid idea. Let's get back to stuff that's not crazy. And she's like, no, I think there's actually something going on. So she spent actually months and months manipulating the detection technique, because she said, I do this all the time for the pathology lab, and it's usually like, tons of virus there, but this is a low-level thing, but it's real. And after we got our techniques down, and a lot of that had to do with eliminating paraffin sections, because the immunology is immunohistochemistry is much better non-paraffin sections, we were able to show that there was clear evidence of viral antigens in glioblastomas, shown in brown here. This is the immediate early 1 antigen in CMV, which is basically an oncogene type of function. And this is adjacent brain on the same section of normal where it's been invading. And then we had multiple different monoclonal antibodies, positive and negative controls. And in the same paper, we did RNA and DNA in situ hybridization, PCR, sequencing, electrogold, gold electron microscopy. So we were pretty confident of our results. And here's just an example that we didn't publish of a GBM that invades into a normal brain. So this is maybe about a centimeter of a pathological slide. And this is the immunostaining for the IE1 protein and the DNA in situ hybridization, which is really prevalent in the tumors. But over here in the normal brain area, there's relatively little, if any, viral. So we found the virus was in the tumors and not in the normal brain. And just to prove the point, I took sections, and this is pretty ugly because it's not presentable for a paper, but over and over again would take sections and do laser capture microscopy, cut out particular areas where we had seen the virus on immunostaining, and then do RT-PCR and sequence it, and yes, there's the virus in there. So we're very confident of this. However, a few people subsequently came out and said, we don't believe it, and it was, it's been a controversial topic. Fortunately, my buddy Dwayne Mitchell came down to UAB where I was at the time and said, this is really cool, and I don't care if you're right or wrong or what, but we're really interested in immunotherapy, and if this is a virus and it's in a tumor and it's localizing specifically to the tumor, we could care less, but we just want to figure out how to do this. So they got the technique working, went back to Duke, and confirmed our findings. And then there were some other people that were pretty skeptical. This guy is a well-known international leader in the herpes virus CMV field, and he went back to his institution and sort of did a blinded study with epilepsy and normal brain and confirmed that there was increased viral genome in the glioblastomas. And here are PCRs from, 12 glioblastomas of all these different viral genes, and they're present in almost all of the tumors. Another guy, Tim Kowalik, who's at the University of Massachusetts, is an international expert on the viral genome, and he independently confirmed and could use Western blots from tumors to show that the IE1 protein, the PP65 protein, these proteins were present in the tumors. And then he sequenced the tumor genes and found that they, in subsequent work, were associated with sort of weird sort of outlier strain, which sets up some of the things we're going to talk about in a minute. So it seemed like there was enough evidence, despite a bunch of negative papers, to pursue, looking into pathogenesis. And as I mentioned, what I was really interested in was to determine, could this virus could the things that we're seeing in the virus, the gene products, the antigens, could they drive the tumor? Just like these EBV things that are well known to drive cancer when they get expressed, like EBNA and LMP1 and those kind of proteins. So the concept emerged that this may be what's called an abortive infection, which means you have an infection, but it's not like a robust infection. It's not producing virus. It's actually just producing proteins so that the virus can sort of survive in that cell but not kill the cell. It's kind of a weird, almost accidental thing that happens in the real world in some of these cancer viruses, that you think of a virus as something that gets in a cell and kills it. Turns out that for EBV in cancer, for instance, it just turns things on that are bad, and it doesn't kill the cell. It actually makes it immortal. So we started looking into the concept of could CMV fit into this paradigm, and could it be, this is a diagram of sort of how cancer evolves in coordination with the immune system. We think that tumors start having mutations and there's immune editing, and that your immune system picks up on antigens that are neoantigens and shuts things down. But after being beaten down for a long enough time, your immune system may say, raise the white flag and just sort of say, I can't handle this anymore. And all of a sudden, those antigens continue to be expressed and go on into a chronic stage. And if you're talking about a virus-associated cancer, you kind of get a chronic indolent inflammation where there's a wound that never heals, sort of an infection that never heals. And then that leads to other downstream things that can promote the malignancy. So one of the first things I noticed was when we added CMV to glioblastoma cells, we got a really big, robust activation of some of the most important signaling pathways that are cancer hallmarks. We found that PDGFR alpha, one of the most important growth factor receptors, is dramatically activated as soon as the virus touches the cell. And that's due to this virus protein that's on the envelope of the virus here called glycoprotein B. It interacts with PDGFR alpha, and it turns on AKT and all these downstream signaling pathways that are classic cancer pathways. And we actually, I'm not a virologist, so I'm a little embarrassed, but we submitted it to Nature, and it got published as sort of, this is the receptor for the virus. And these virology guys are like, hey, how did you figure that out? It was kind of a total accident, but I'm glad we could contribute. But what we find is in the tumors, this viral glycoprotein is expressed on the surface of the glioblastoma cells. And these cells also have hyperexpression of PDGFR alpha, so that's an autocrine signaling pathway that drives tumor proliferation. And we proved that in vivo, and I'm not going to go into that. Around the same time, one of my colleagues in the Netherlands found that one of the viral gene products called US28, which is a chemokine receptor that the virus produces, gets expressed during infection. And if you put that chemokine receptor into a tumor cell, that viral protein alone will drive dramatic upregulation of the proliferation of the tumor. And these are actually GBM cells, where the tumor cells have dramatic increased blood vessel formation due to increased VEGF production. So the pathway is sort of activation, sustained activation of this thing, driving pathways downstream that increase inflammation and expression of angiogenesis. And another group actually put this gene into a mouse model of colon cancer, and that one viral gene alone caused colon cancer in mice. So we went back and looked at our tumors, and we found that, yes, these GBMs in patients have this viral protein expressed, and here's a RT-PCR. I guess about half of them we could amplify the gene product, and we showed that if we activated these gene products in tumor cells, we would get a similar upregulation of proliferation. I'm just kind of giving you a background of some of the things we've done, and then I'll get to the sort of the importance, I think, in terms of patients. Another thing that was really fascinating, and you just heard a talk about cancer stem cells was that when we were growing neurospheres of these tumors in patients, we started staining them for the viral proteins, and we found that the virus seemed to really be highly expressed in the glioblastoma cancer stem cell subset, which may be less than 1% of the total tumor compared to the rest of the tumor cells. This is a very slowly dividing virus in vivo, and it actually turns out that these glioblastoma cancer stem cells are actually much more slowly dividing than the rest of the tumor cells. So, the hypothesis is these are sort of the perfect host cells for the virus to sort of live. And my sort of simpleton's analogy is kind of like, if you can sit down in a room with a sofa and a remote and just eat Cheetos all day, it's like the virus is set in these cells. It just like hangs out. The cell does all the work. It has the perfect viral life cycle for the virus, and the virus seems to like it. So we started exploring this, and you can see here, these are some neurospheres that we've immunostained with the viral IE1 protein in green, and it co-localizes in these tumors right out of a patient with nestin, which is sort of the prototypical glioma stem, one of the undifferentiated markers. And then there were some other things that co-localized with, like SOX2, which is one of the driver gene products for cancer stem cells. So this was really fascinating to me, because then I started talking to Liliana, the postdoc or the PhD that I worked with on this, and said, is this a chicken or egg thing? Does the virus get turned on in these stem cells, or is the virus making the stem cells stem cells? And so we started doing some experiments, and this is just a FACS sorting of a glioblastoma, where we showed that there was indeed a correlation of viral gene expression and glioblastoma stem cells from a patient that were positive for the IE1 protein, suggesting there's some preference of the virus expression in these glioblastoma stem cells. Oh, and this, around this time I was talking to Amy Heimberger at MD Anderson, and her lab sort of started looking into this, and they corroborated that, because when they did a similar experiment where they took glioblastoma tumors and sorted the stem cells with a CD133 marker, and they found that the high expressors of CD133, they could have increased evidence of this viral protein PP65 or the IE1 protein. Interestingly, they found that there were high levels of the viral proteins in the macrophages in the tumor and the tumor cells, but not in the lymphocytes. And then if they co-cultured the viral infected glioblastoma stem cells with immunological cells, what they found was that these glioblastoma stem cells secreted a viral chemokine called IL-10, a viral IL-10, which was very immunosuppressive. And so the T cells that were co-cultured with these virally infected tumor cells started having very high levels of expression of CTLA4 and PD-1, which are the sort of the prototypical checkpoint inhibitors that make T cells not function. So they kind of did a lot more than we had in that with these tumor stem cells, but they showed that they are associated with the viral gene products, and they can pump out things that shut down the immune system. So Lily, who's the first author of this paper, and I started looking into this concept with all of our folks in the lab at the time and some collaborators from other institutions. Long story short, we had some really remarkable findings. We found that, if we were to take glioblastomas and we were to, sometimes look for the viral proteins or the RNA of the virus, we couldn't see it in a sort of a quick and dirty Western blot or RT-PCR. But if we sorted out the glioblastoma stem cell subset, which may be only a fraction, 1%, we would often find it. So the virus seemed to be really harboring, staying in those stem cells. What was actually most striking, though, is we would grow in multiple occasions, we would grow the glioblastoma stem cells as neurospheres in the lab. And then we made a siRNA actually two siRNAs that knock out this one viral gene, IE1, which is the one we thought was sort of the driver of the virus and maybe a driver of the stem cell biology. And if we treated these glioblastoma stem cells right out of a patient with something that knocked down that one viral gene, we could shut down the glioblastoma stem cell growth in culture. And that's evidenced here on a Western blot. Here are the cells before treatment. Here's after we knocked out the viral gene, so the IE1 gene is gone, or protein is gone. But also we showed using a blot that has all these dark little spots here are proteins that are related to cancer stem cell biology. You can see after knocking that one viral protein out, almost all of them are shut down, and SOX2 is a really important one of those. So, and if you take these stem cells and you just let them grow in culture, this is what happens. They get, more and more cells. However, if you add one, two, or both of the knockdown siRNAs, you can essentially shut down these tumor stem cells right out of a patient. So this is pretty striking finding. And another thing we notice is this virus does not integrate into the DNA of the cell. How long do I have to go here? I don't want to go too long. Oh, I'm already got till 10:20. Okay. So the virus doesn't integrate in the cell. So what we found is if we grow these tumors in culture, the virus would often seem to be gone after 2 or 3 passages, which is consistent with what happens in Kaposi sarcoma virus, which is another herpes virus that causes cancers in AIDS patients, as well as in some cases of Epstein-Barr virus-related tumors. Viruses don't like to be in tissue culture. But in those cases where we could grow the neurospheres after the virus had gone away, if we infected the cells again at that point, they grew like crazy, as if we were adding, massive amount of growth factors or something to these neurospheres. If we did that and at the same time gave the siRNA to block the viral gene, they didn't grow. So it seems like what's going on, at least in a simple concept, is that these tumor neurospheres of the CMV-infected glioblastomas are being supported by expression of a viral gene that may drive stemness of the stem cells. And we actually showed in this paper that there's a microRNA called microRNA 145 that actually interacts between the viral genes and SOX2, OCT3 and 4, and NANOG, which are all the stem cell drivers. And furthermore, we took this system and we put this one viral gene, IE1, we put that gene into a mouse model of brain tumor. And just by adding that one gene into the mouse model of the brain tumor so that it was expressed in the cells that turn into tumor cells, the tumors grew much more rapidly and they had much more stem cell-like properties. So it's a pretty cool concept. Around that time, I put together a little small meeting thanks to ABTA in Washington, D.C., and invited my buddy Nino Chioka, and he does a lot of viral therapy research, and he had a student, Rick Price, who's actually applying here for a spine fellowship this year, who was a medical student, MD, PhD student, and so they went and got a mouse model of glioblastoma from Louis Parada, who's now, I think it's Sloan Kettering, and these mice develop spontaneous gliomas that are usually grade 3 and then turn into glioblastoma. And so what they did was they infected them with CMV at the first day of birth. And just as a little background, every species has its own CMV, but mouse CMV is very analogous to human CMV. It just shows you that evolutionarily this virus has been around before mammals existed, basically. So what he found was when you infect these mice with murine CMV, they get this sort of widespread infection. It goes away just like in a normal person. And then when the tumors start growing after a month or two, they get infected. And not only do they get infected, they start expressing viral gene products, and those viral gene products drive a lot of the STAT3 signaling pathway things, and they showed that in this paper that there's high levels of phospho-STAT3 in the tumors, and the tumors grew bigger and the mice died more rapidly. So the virus was a kind of a cofactor in driving more aggressive, rapid death. And I got a grant from the NIH to look into this further, and kind of still working with Nino, and Sean Lawler is really the PI that's doing this work at the Brigham and Women's. But we have a paper under review right now. It's pretty exciting because basically Sean has repeated this in 2 different more syngeneic mouse models. One's called GL261 and another one's called CT2A, I think. And in any case, it's repeatable consistently so that if you give the virus to the mice, they get the tumors get infected and the mice die more rapidly. But what was fascinating is when they started, looking at MRI, they showed the tumors were growing bigger. But not only that, the tumors, when they analyzed them, they were much more invasive and infiltrative, and they seem to have this. You can't really see this, but there's much more CD31, which is more blood vessel formation, and there's much higher Ki67, which is proliferation rate of the CMV-infected tumors. And when they drilled down on that, they found that not only is there more blood vessels, there's much greater blood vessel density, and then they did MR blood flow studies and showed there's a lot more blood flow to the tumor. So that sort of led to the second part of the project, which was, what's going on? And what's fascinating is it turns out, just like in the human tumors, we have the viral proteins PP65 and IE1 expressed in the tumors, but then there was this dramatic increase of these blood vessels, and these blood vessels had a marker called NG2 that was highly expressed. And NG2 can be a molecule that marks oligodendrocyte precursor cells, but if it doesn't have the oligodendrocyte component, then it usually represents pericytes, which are the little cells that sort of help line new blood vessels. So basically, this study showed there's a dramatic, shown in this figure, increase in pericyte formation in these tumors, and they went on to show that that's due to the tumors that are infected with CMV are pumping out platelet-derived growth factor, and that seems to be causing a massive influx, possibly from the bone marrow, of cells that then turn into blood vessel cells in the tumor that are infected now with the virus. So there's this, infection in the blood, in the bone marrow stem cells. There's infection in the tumor. It's all kind of one organ, sort of, so to speak. And interestingly, if they block that growth factor with an antibody, they can shut down this whole increased proliferation of the tumor with vascularity. And based on some stuff that was another part of our grant, the question is, can we give an antiviral drug? And what's really exciting is, in this setting, if you give an antiviral drug, you don't see all of this stuff going on, and you can shut down the effect of the virus on promoting the glioblastoma in this mouse, and they live the same as without the virus. So pretty exciting that there's now some mechanism and maybe a suggestion of a role in a mouse model for why an antiviral drug might work. So one thing that I'm really kind of obsessed with now is, why has it been so hard for us to sequence this virus? And we're starting to get some hints, and I'm working with some brilliant people at the Institute for Systems Biology who just had a landmark paper in a journal, Neuron, where they have discovered that in Alzheimer's there are herpes virus infections that had been unbeknownst to other investigators. So we're working with these guys, and we've got some very interesting preliminary results. So my questions are, what is this? Is this normal CMV? And we have some reason to believe that it may be some kind of weird strain. And I guess one analogy that we could sort of work off of is, when Harold Zerhausen first proposed in the 1980s that HPV caused cervical cancer, people said, you're nuts. When he gave his Nobel speech a few years later, he said, well, it's not just HPV, it's just these two strains of these three strains of HPV. So maybe we're dealing with something that's not what everybody thinks it is. So, and the other thing is, why doesn't the immune system function in these patients, and why, how is it possible you can have somebody who has this persistent infection with viral proteins expressed. And I think that leads to the next question. Let's say you could target this virus. It would be the perfect vaccine target for immunotherapy. If all the tumor cells are infected, or especially if the tumor stem cells are infected, and you can mount an immune response, maybe you could wipe out the tumor. So the first thing that, I'll just go through a couple of papers that are interesting. We have a colleague, Cecilia Soderberg-Noclair in Stockholm, who came here once and gave a talk, and we've been very fortunate to steal one of her top scientists who just started in my lab a couple of weeks ago. Basically, you look at the immune response to CMV in these patients, and it's kind of weird. A lot of them are not seropositive, which is sort of the gold standard to say that they've been exposed to the virus. And they showed, well, some of them are seropositive, but not to normal CMV. They're to some clinical strains. And then those that didn't seem to be positive, they had T cells that had seen CMV. So something's weird about their immune response to CMV in general. And then this is a really good paper from a group in Norway that drilled down on this even further. They actually took glioblastomas from patients, took the T cells out of the patients. They showed that the tumors were positive for CMV, and then they showed that there was a significant increase in seropositivity to CMV in the glioblastoma patients, with a 4.0 odds ratio. But what's most interesting is they said that while these people did have increased IE1 and other T cells specific to these viral antigens, these T cells just basically had very high levels of CTLA1 and PD1, which are the checkpoint inhibitors that block their function. And so even though these patients had these T cells that were trying to get these CMV antigens in the tumor, they were non-functional when you tested them. So they're just not working. So it's like the immune system is there, it's circulating in the tumor, but it's shut down, it's not functioning. So that allows the virus to persist. And so now I'm going to kind of transition into, what are the possibilities that we could leverage this understanding to cure patients. And the first hint of something kind of cool going on was, we have folks we did the DC vaccine. We have folks from DC Vax here. So Linda Liao was one of the main initiators of that concept. And so when Linda was early on in her studies, she, basically you take a glioblastoma, you take the proteins, you put them in the dendritic cells, and you let the immune system go after whatever's in there to attack the tumor. She started looking, after we published our stuff early on, at some of these patients, and she found one patient, and they wrote it up in New England Journal, just kind of checking, does CMV activation occur in this setting? And this patient, they vaccinated with their own tumor proteins, and they saw a dramatic increase in the T cell response to one of those proteins, PP65. Interestingly, this patient had a mesenchymal form of GBM, and according to Linda, this patient is cured. They had an aggressive GBM and unmethylated mesenchymal, and it's been 14 years now, and there's no tumor. That's a coincidence, maybe or maybe not, but that sort of synchronized with what the Duke guys were thinking about, and some other people at the same time. And so Dwayne Mitchell, as I mentioned, went back to Duke, and he started taking T cells from patients and stimulating them in a dish with some of the viral proteins and saying, if I stimulate these T cells in a dish against this one prototypical antigen of CMV called PP65, what would happen if I then let those T cells go after the patient's own tumor cells? Will it kill them? And what he found was, yes, that actually did happen. As controls, he used, I can't see all these things, but he basically vaccinated the T cells or exposed them to all different antigens. One is from the flu, one's survivin, one's some other sort of standard things, and then the ones up here are PP65, and this is killing of their own tumor cells. So when you take someone's T cells, you stimulate them against these viral proteins, they can actually have the capacity to get turned on. And that's shown here because when you pre-stimulation, this is a T cell response. Post-stimulation, you can see on the FACS sorting, there's a dramatic increase in the ability of the cells to secrete gamma interferon, which is sort of, by definition, activation of the T cell. So this was kind of a theoretical, yes, these tumor cells do have the protein, and here they showed it on a Western blot. The tumor cells are positive for the protein. The T cells from the patients don't work, but if we stimulate them, we can get them to reactivate and then go back and kill those tumor cells. So at the same time, Raj Khanna in Australia, who is one of the leaders in vaccinating against Epstein-Barr virus-related cancers, started looking into this, and he sort of took a similar approach. He said, can I take T cells from a patient, can I mix them with a bunch of viral proteins, peptides, little segments of the viral proteins, give them, cytokines to rev up the T cells, give them back to the patient, and see a response. And so he did a clinical trial with, I think, 11 recurrent glioblastoma patients, and he showed that in those patients that he could induce expansion and activation of those T cells after exposure to these peptides, that in almost all cases the cells had increased expansion, increased expression of cytokines to these peptides. And then, although it was a tiny little recurrent glioblastoma clinical trial, the median survival was 400 days, and some patients seem to have had, down here, that, I don't really have a pointer, but that bottom thing, there seemed to be about a, really long-term survival, like 3,000 days for some of these patients, suggesting that maybe reactivation of their T cell response to CMV shut down their tumor. Around the same time, my colleague in Sweden, who I mentioned, did a quick and dirty clinical trial, which was unfortunately designed horribly, because the plan for the trial was, because she didn't have enough money, was to do a prospective randomized trial, give some patients valganciclovir, which is an oral antiviral, and give some patients placebo, and then see if there's a difference at 6 months. Anybody who treats glioblastomas can tell you that at 6 months, you're probably not going to see a whole lot, because most people sort of, after radiation and chemo, they kind of just sit still for a few months, and then things take off. Then she reanalyzed the data after following those patients, and she found that the patients that did receive valganciclovir had striking overall median survivals. And she published this as a letter to the New England Journal, which was received with a lot of skepticism, because any sort of neuro-oncologist would say, you can't go back for a negative study and then reanalyze the data, blah, blah, blah. Nevertheless, when you really parse out what's going on, it would be hard to cherry pick these patients. They were all the same unmethylated percentage and everything else. So she's continued to do follow-ups, and there's some unpublished data that she has that's quite striking. Now, the median survival in the patients was something like 40 months for these GBM patients that were on antiviral drug. And then at the same time, the Duke guys did a vaccine study. I think I have it better. Oh, let me just do this one first. Another study, which doesn't provide much information, was done at Vanderbilt, where they gave, for recurrent GBM, either Avastin or Avastin plus valganciclovir, and there was a statistically significant improvement in survival. I'm just throwing that out there. It was just kind of a small study. But this was quite impressive. So the Duke guys, Dwayne Mitchell and John Sampson, basically said, okay, let's really go after this PP65 target, because it's one of the most robust viral antigens. And so they took patients' dendritic cells, they pulsed the RNA of the viral gene into them. Those dendritic cells were then given back to the patient, and basically, the hope was they would say, immune system, go after this viral protein. And what they found was pretty striking, that in 13 patients, the median survival was something like, 4 years. And they did a follow-up study, which was similar. So they vaccinated these patients with a vaccine against the CMV PP65 from their own dendritic cells. And I'm not going to go through all the data, but basically, this shows the patients whose T cells seem to get activated after the vaccine, and it's broken down into the black spots are people that live less than 40 months, and the hollows and the clear spots are the ones that live longer. So the median survival in this study was something like almost 50 months, 45 months, which is pretty striking. So they actually said long-term survival, kind of unexpected. And it's a phase 1 study, but there was a correlation between response to the vaccine in terms of T cell activation and survival. So it's kind of it's not a causal association, but it does hint that there is some kind of association. And then they had another trial they did recently that, I won't belabor the point, but basically they showed that in patients who had improved survival, there's definitely an increase in the ability of the T cells after vaccination to have robust responses to 3 really important cytokines, interferon gamma, TNF alpha, and CCL3. And it sort of plots out that their survival and their response to the vaccine is associated. So that's led to, that has led to some more interest, and this is just from clinicaltrials.gov. I guess there are 8 active trials now looking at CMV vaccination. Most of them are at Duke, and 3 of them are enrolling. Different ways of doing it, vaccinating a vaccine with dendritic cells or peptide vaccines. I've actually tried to get into this clinical trial and world, but it's hard to do unless you're at a big place like Duke. So in summary, I believe this virus, still a lot of stuff to do, but I believe this virus is involved in these tumors. And the goal of our lab now is to really drill down on sequencing to figure out what viral strains exist, figure out which genes are expressed, and then use mass spec and other techniques to figure out the proteins, and then figure out which are the best targets. We don't even know if these are the best targets so far. And then consider vaccine trials in combination with checkpoint inhibitors to make it more robust. So the people that have done most of the work are shown here, and I've been supported by NIH and Ivy Foundation and a few other foundations and Susan McGregor Fund, and I want to thank them all so much for their help. Thank you.